Today we will be continuing the discussion over cellular metabolism. Specifically, we will be talking about coenzymes that are involved in redox reactions. Now remember that redox reactions are the main types of reactions that your body does during metabolism in order to do energy production. So what are the two main coenzymes that are present in metabolism? The two main coenzymes are going to be NAD plus and FAD, right? So when your body does reactions, okay, a lot of the energy is produced, but it's kind of trapped, right? So the energy that is produced in these metabolic pathways are produced as NAD and FAD. But you can't really use that in this form, right? So this is kind of like an intermediate. So you can think of intermediate. It's kind of like making cookies, right? So you make the batter, you follow all the step-by-step procedures, but you have the cookie batter. Are you going to eat the cookie batter raw? Personally, I would. I mean, like the process stops there. I eat it. That's cool. I live my life risky, okay? But some people, a majority of people, say, well, a lot of this energy, a lot of the product is trapped within the batter. It's trapped within the NAD and FAD. So I have to put it in the oven, I have to do more reactions in order to get my cookies, in order to get some ATP, okay? So we have to do more reactions to these guys in order to get our ATP, which is the overall product of the metabolic pathway, okay? So what happens here is that we have to do kind of this long word, but essentially we have to do dehydrogenase enzymic reactions. So dehydrogenase, hydrogenase. So we can break this down. We could say D means to take away, hydro is hydrogen, and genase means something with an enzyme. Dehydrogenase enzymic. So that just shows that, yeah, we are going to be doing something with enzymes. Of course, reactions. Okay, so what does that do? Essentially, whenever you do dehydrogenase enzymic reactions, you're going to be reducing the NAD and FAD. So what does that mean? Well, remember when I was talking about oil rig, right? We have oil rig. Well, it means that the oxidation is loss of electrons, and whatever is oxidized is going to be reduced. Rig means reducing is gaining electrons or hydrogens. You'll see why I'm talking about hydrogens in a minute. So whatever is reduced is oxidizing agent. Sorry. So this is oxidizing agent. Okay. So when I say that, oh, we're going to be reducing NAD and FAD, our main coenzymes, what am I saying? I'm saying that since we're reducing them, we're going to be gaining something. We're going to be putting hydrogens into this reaction, into these coenzymes. Sorry. So we got this hydrogen from a different reaction. We're going to take away that hydrogen from a molecule and shove it into the NAD or FAD. This means that these guys right here are going to be oxidizing agents. Okay, because they are going to be reduced, right? They are going to be reduced. So how does that look like? Well, you're mainly going to be working with the hydride ion. So you remember hydride in OChem? It's like a hydrogen with some lone pairs, okay? And it's negative, right? Personally, I just like working with like a negative hydrogen. Makes it streamlined, so that's what I'll be using, okay? So what happens here is that NAD, okay, NAD is nicotine amide something, but it's not important for you to know the name, is going to be attacked by the hydride. So the hydride is going to attack it. So this is going to be reduced. This is an oxidizing agent, so we just say oxidizing. So that was reduced. And we know that the hydride is going to be losing its negativity in this case. So that means that it is going to be a reducing agent. Reducing agent. And what you're going to get is NADH, right? And similarly, if you did it with FAD, you would get FADH, right? So why is that important? Well, both FADH and NADH are going to go into your membranes. So let's use an example. So it's going to go in your membranes and create kind of like ATP. How does that happen? To do that, I have to first clear the board. Okay, so that's always fun to do, right? So in your membrane, specifically the mitochondria, you have these little complexes, right? So textbooks are going to draw them out as squares, and I'm going to do that too because I am a master at drawing squares. Okay, so we're going to be doing that. So this happens in the mitochondria, and this is complex 1, this is complex 2. You can think complexes as little factories. This is complex 3, 4, and this is 5. 5 is going to be called ATP synthase. Okay, and they're inside the membrane of the mitochondria. Okay, so they're within the membrane. Okay, so this is outside the membrane, and this is inside the membrane. And what happens here is that NADH is going to essentially touch this complex, is going to generate hydrogens, okay? And well, you don't really need to know the amount of hydrogens that it generates, okay? But complex 1 is essentially connected to complex 3 and 4, right? And so whenever NADH touches complex 1, it generates hydrogens. This one also generates hydrogens, and this one also generates hydrogens, okay? And so you're having a buildup of hydrogens in this membrane. FAD, specifically FADH3 touches this and generates hydrogens as well, okay, via 2 and 3. So that's not really important, but what's important is that we're using these intermediates, okay? So we essentially reduced our oxidizing agent, and then we're pushing it to a different reaction. So we're using our intermediates to create a hydrogen gradient. These hydrogens then go into ATP synthase, which is complex 5. A phosphate comes in as well to help out, and out comes ATP. Okay? So, like I was saying, a lot of this energy that we make during these these redox reactions are going to come out as coenzymes, right? So NADH, FADH3. Who cares? I don't care. Well, you should care, because NADH and FADH3, which were created by reducing your oxidizing agents via the process of dehydrogenase enzymic reactions, well, they actually propel this to happen. Without these molecules, right, these coenzymes, you're not going to be making ATP, you're not going to be feeding your muscles, you're not going to be able to function. Your organs would shut down and you would die. So without these processes, without having redox reactions, you wouldn't be alive. You wouldn't be able to enjoy Cheetos. You wouldn't be able to pet pigs. So that's why we're studying it. That's why it's so important, okay? So this is actually a key step in glycolysis. We may cover that later on, but that's essentially what you need to know, that these guys were formed via dehydrogenase. Dehydrogenase enzymic reactions, okay? And that's just a fancy way to say, hey, somehow get a hydrogen, shove it into NADH or FAD, create a different product, use that product, and create ATP. That's all it's saying, okay? And another key factor that you need to know usually is that oxygen plays a huge role whenever you're doing dehydrogenase enzymic reactions. okay? Because you're going to have oxygen over here, and essentially oxygen touches this as well, and it produces water, okay? So you're essentially oxidizing, you're losing electrons, right? Because the amount of the amount of oxygen's decreased, so that was an oxidation. So this is a reducing agent right here. Without the use of oxygen, you wouldn't have this process of making ATP. So in order to get oxygen, you have to do an aerobic pathway. So you have to do the aerobic pathway to use oxygen, okay? So aerobic just means air, right? So if you're doing aerobics, you're going to be running, you're going to be doing jumping jacks, you're going to be breathing, okay? So O2, or oxygen, is, let me write it here, O2 is oxidized. So it lost electrons, so it's a reducing agent. Reducing agent, very important. So you can have your NADH and your FADH3, right? So that can be like your cookie batter. That's great, but you can't have cookies, ATP, without having an oven. If you have no oven, oxygen, you're not going to be doing a reaction completely, okay? You just have your ingredients, but you have no product. We will now be talking about group transfer reactions. So group transfer reactions are pretty common. Group transfer reactions. Of the group transfer reactions, the most popular one is called phosphorylation. So most common is phosphorylation. Phosphorylation. Okay. So before we talk about that, remember in the last slide when I was talking about ATP synthase, the complex 5, and I said, oh, a phosphate comes in and helps out. Well, technically, the phosphate came in and phosphorylated some, mechanisms and produced ATP. So we actually transferred a group of phosphates into a molecule. That's what we're doing. So essentially, group transfer reactions are just reactions that literally transfer a group onto a molecule. For instance, we have this example. We have a cute little glucose. Okay, so it's a simple 6 carbon sugar. Okay, and it's going to be, you're essentially going to add a phosphate onto it. Okay, so when does that happen? Well, it happens when cells, let's say that you just ate some Krispy Kreme donuts, okay? My personal favorites are raspberry filled, right? And let's say that I eat a raspberry filled jelly donut. I eat it, it goes into my intestines, and my intestines are going to break it down and absorb nutrients, okay? So it needs glucose, it needs some carbs, so it starts breaking down the glucose and starts shoving it down the cells, right? So we have some cells right here, and then we have glucose, let's call it G. So glucose is going to enter the cell, and then inside the cell are going to be ATP molecules, okay? Now, ATP is going to do a group transfer reaction and essentially just, cut a phosphate group. So it wants to be favorable. We know that if you break a phosphate bond, it's going to release a lot of energy, so that's good. And this phosphate is going to say, hey, I'm attracted to this oxygen right here. So what happens here is that the oxygen is going to grab a phosphate. Why does it grab a phosphate? Well, it grabs a phosphate because when it does that, it's not going to be able to exit the cell. Okay, so in this example, group transfer reactions of the phosphate group onto the glucose is going to prohibit the glucose from exiting the cell. And that allows the cell to kind of absorb nutrients, do reactions with the glucose, incorporate it into its metabolism, right? So you don't want your fuel source to be, zipping right through you. You don't want that. So we phosphorylate the glucose in order to keep the glucose inside the cells for us to, use the glucose for metabolic pathways. So that's pretty common. It's usually tested. So just know that group transfer reactions, you're just essentially adding a group onto a different molecule. The most common group transfer reaction is phosphorylation. Now, what actually drives phosphorylation? You're saying, okay, well, how do we cut the ATP? Are we going to be using like an enzyme or something? And the answer is yes. Usually the answer is yeah, we're going to be using enzymes. Specifically, we're going to be using the enzyme kinase. Okay, so kine usually means motion. Okay, and ase just means that it's an enzyme. So literally it means motion of enzyme, or enzyme in motion. Okay, and what it does is that it just sets the reaction into motion. It's pretty funny if you think about it. So kinase says, okay, we have some glucose coming in. It's raspberry filled. I love that. We're gonna put into this reaction. We're gonna put this reaction into motion. I'm gonna cut the ATP, make ADP, and a phosphate. That phosphate, right? Because we make ATP and ADP plus a phosphate ion, right? So it says, okay, well, kinase says, now that I cut ATP and made ADP and phosphate, I'm going to move the phosphate onto the glucose. And when that happens, the glucose is not going to be able to get out the cell, and we can use that glucose to our advantage. We can now use it for metabolic pathways. So essentially that is the group transfer reaction. We will now be talking about isomerization and rearrangement reactions. So we're going to be talking about isomerization. Okay, isomerization and rearrangement. Reactions. Well, without using big words, isomerization is when you have the same formula. Okay, let's just make up something. Let's just say kind of like this, right? Okay, so that's CH3, CH2, CH2, CH3. Okay, isomerization is when you just make a different compound using the same amount of carbons and hydrogens or whatever you have, right? So I have 1, 2, 3, 4 carbons. I have 4 carbons. And how many hydrogens do I have? 3, 6, 7, 8, 9, 10. I have 10 hydrogens. Okay, so can I make a different compound? Yeah, I can. Right, I can have that, and then boom. Right, so I still have, let's see, 3, 6, 9, 10. I have 10 hydrogens and 4 carbons. Okay, so that is isomerization. It's when you make a different compound using the same amount of atoms, right? So they have different properties, they do different things. Depending on if you had stereochemistry, they can reflect light differently. So that's what we're doing. So in these reactions, we can actually create isomerizations, right? We can do isomers, right? So here's an example. We have, well, first of all, this doesn't change, so we can just ignore it, right? So we have 2 carbons right here, and we have 2 carbons, cool. We have 2 oxygen, and then we have 3 hydrogens. Oh, we have 3 hydrogens there. So let's just focus on the top. We have 3 hydrogens, 2 carbons, 2 oxygens. Okay, over here we have let's just do 3 hydrogens, 2 carbons, and 2 oxygens. So we didn't remove any atoms, but we did move some bonds. So we actually moved around some hydrogens, and we got a different compound. Okay, so in this case, this is called an intramolecular hydrogen rearrangement shift. So it's kind of weird, but it's like intramolecular. So that means it happens within one molecule. That's what intra means, right? So interactions, inter means between 2 different things. Intra is between one thing. Intramolecular hydrogen, let's just put H, shift. Okay, so literally you're just shifting the hydrogen. Okay, so that is one type of reaction. The second type, and that's the one we're going to be covering in this class, is, well, an intramolecular rearrangement of a group, specifically a functional group. So intramolecular rearrangement of functional group. Now I want you to look at this this glucose atom, this glucose molecule. So notice that nothing else changed, right? Everything stayed the same except for this guy and this guy. So what did we do? Well, I essentially popped this guy out, took a hydrogen, made the oxygen get that hydrogen, and then got this oxygen and connected it to this phosphate group. That's all I did. So technically, we rearranged something, right? I'll go over to again. We popped off a hydrogen from an oxygen. Then we took off a phosphate group. We got that phosphate group, put it to the oxygen that had its hydrogen removed, and took that hydrogen and put it on the oxygen that had the phosphate group removed. So we just rearranged it, okay? So that happened between one molecule, and that is called an intramolecular rearrangement, okay? So if on the exam they're like, okay, what does isomerization and reaction and rearrangement reactions do? What do they do? Well, you just say they get one compound, they either do hydrogen shifts or rearrangement of functional groups and create a different molecule. So for instance, this one right here is an aldose. We'll cover that later when we're talking about carbohydrates. And this one is a ketose, right? So aldose and ketoses, they have different properties. They do different things, yet they still have the same molecular formula. We just did an isomerization, okay? So that could allow you to convert to different products, different reactions. Remember when we were talking about catabolism and we were saying, well, there are 3 steps, and the first step is to break down a macromolecule into its building blocks? Well, this is what happens, right? So let's say it's 2 o'clock in the morning, you were hungry, so you ate a stick of butter, right? How does your body break down the fats so you can use it or store it? Well, your body is going to be using some types of reactions called hydrolytic reactions. Hydrolytic reactions. Okay. Hydro means water, lytic means cut. So we're using water to cut some macromolecules into their building blocks. In lipids, you're going to have triglycerol, right? So that's just a glycerol with 3 fatty acids together. Now, the glycerol and the fatty acid are linked together by an ester bond, and that ester bond is quite fragile. So water can just mosey along and break down that ester bond, okay? And what happens now is that you have a 2,3-diacetyl glycerol. Essentially, we just use 1 water to get 1 fatty acid. So how many waters do we need to completely hydrolyze? It sounds weird. Hydrolyze a triacetyl glycerol. How many waters do we need? We need 3, right? So it's 1 water per fatty acid. So this is called ester hydrolysis right here. So this is ester hydrolysis. And it should be pretty straightforward. It's in the name. So we break an ester bond with what? With water cutting. We just use water. What about for amino acids? Okay, so you use a chicken leg. How do we break down that muscle, that protein, into usable compounds? So water is going to look at this part right here. So you remember when we were making peptides? I said, well, we can link peptides together, but when we link them, we're going to have to expel water. So remember, if we had alanine and we add another alanine, when they come together, they're going to make 2 alanines, right? And then they're going to shoot out water. But now, after like a cajillion lectures, that water is going to get its revenge, right? Because it's mad. So it's going to come back and say, nope, I'm going to cut you. And now you're just going to produce alanine and alanine, right? So what happens here is that it goes to the peptide bond. So this is the peptide bond, specifically the peptide bond. Okay, so on the exam it says, where does hydrolysis occur in lipids? Oh, that occurs in the ester bond. Where does it occur on amino acids? Oh, it occurs on the peptide bond. Okay, so between the C terminus of a protein, if you want to get technical. So it does that. The water comes into this peptide bond, it breaks it down, and now you have this individual amino acids. Okay, so I don't really like this figure, but you just have individual amino acids, right? So there you go. This is called amide hydrolysis. Hydrolysis right here. Ooh, that was pretty bad. So hydrolysis. There you go. Okay. And now what else? Right here, we're going to be having a glycosidic bond hydrolysis. So this is pretty dumb, but this is glyco. So glyco usually means sugar. Okay. Glycosidic bond hydrolysis. Okay, so there you go. Essentially, this right here, whenever you link two simple sugars together, you're going to have a glycosidic bond, right? So that's just essentially, let me write it in a red. So before you even linked it, you had, let's ignore everything. You had this run right here, okay? An oxygen and hydrogen. And you also had it over here. So R is just, a random group of carbons. It's just the other part of the glucose. I'm not focusing on that part. I'm just focusing on the interesting part over here, okay? So over here you have the oxygen, specifically hydroxyl. And what's going to happen now is that whenever you want to link sugars together, say you just ate, 3 pints of ice cream and you're watching Grey's Anatomy or something, I don't know. So in ice cream, specifically lactose, because ice cream is made of milk, lactose is in milk, these hydroxyl groups are going to be bound to each other, right? That's how you link glucose. So that's what you're seeing over here. The hydrogen left over there and another hydrogen left over here. So whenever you lose hydrogen, you link glucose together, right? And so now water can come in and it could get its revenge, and it says, you know what? I don't like lactose. I'm going to come in here and I'm going to break this bond and I'm going to mess everything up, okay? So that's what happens. So notice that we have the hydroxyl group on the upside, and we also have a hydroxyl group on the downside, okay? So that's all that happens. On the exam, if they ask you, if you want to break the bond between two glucose atoms or two sugar atoms or whatever, where does that breakage occur? You're going to say the glycosidic bond. Right here, this is the glycosidic bond. Glycosidic bond, okay? And that's essentially any bond that you want, essentially a glycosidic bond is a bond that bonds any glucose, right? So it could be fructose, lactose, galactose, mannose, it doesn't matter. It's just a hydroxyl coming together, okay? Another type of reaction that might occur during catabolic pathways, okay, are the non-hydrolytic cleavage reactions. So these are non-hydrolytic cleavage. In this case, cleavage means breakage, or we're going to break something. Cleavage reactions. So the most common bond that is cut during those reactions are carbon-carbon bonds. So carbon-carbon bonds usually are targeted. Okay. So as we can see over here, we just get rid of this bond right here. We have kind of like this little tidbit, which is over here. And then we also have what we cut off. So did I use water to do that? No, I did not use water to do that. Okay, so that's a non-hydrolytic cleavage reaction. So sometimes whenever you do these types of reactions, you actually get a double bond. Okay, and that is most typically with enolates. So this is a kind of like a subsection. So enolation. It's essentially when you dehydrate the molecule. So over here you can see that in this picture there's a hydrogen highlighted and also a hydroxyl group highlighted. Whenever we do a non-hydrolytic cleavage reaction, in this case, in this example, we dehydrated the molecule, so that means we generated water. And then look at this cool stuff. We actually made a double bond. So double bond created. Now whenever you dehydrate a molecule and you make a double bond, that is called an enolate, okay? And we call that process enolation. Finally, the last type of reaction that catabolism uses during metabolic pathways are going to be bond formations using the energy from ATP. Okay, so for instance, before we get into it, I have to explain to you how this works. Normally in reactions you have a carb anion, right? And it's going to attach itself to the carbonyl group of a molecule, right? And the reason why it wants to do this is because this carbon right here is going to be stabilized by the electronegativity of this oxygen. So it is stabilized by the induction. So stabilized by the induction effect. Okay, so there you go. So that's why carbanions want to actually attack this carbonyl group with a hydrogen, and it's going to be more stable. So this one right here doesn't want to be like that. It takes more energy to maintain that form. And this form right here is stabilized. It takes less energy to maintain. It's just a lot better, right? So the carbanion is going to do that. Typically, you see this with ketones. You see this with ketones. You see that with esters as well. Esters and CO2. So now we have a couple of examples over here. So over here we have pyruvate, which is typically a product of the anaerobic pathway, right? So we have pyruvate, we're going to be mixing it with carbon dioxide. Now notice that carbon dioxide is kind of like a carbanion, right? Is going to be using some of the energy from ATP to create a bond between this carbon right here, because it wants to be stabilized by the induction effect, okay? So it uses ATP to make a bond, right? And this creates ADP and a phosphate ion, okay? So that's all that's doing. That's all that's happening. So how do we do that? So do we use like an enzyme or something? We do use an enzyme. We actually use two types of enzymes depending on the reaction. Specifically, we use the ligases, and we also use synthase. Okay, so synthases, they just make molecules. Okay, so you're synthesizing molecules. Here we have another example. Specifically, this is going to be a ketone, right? So as you can see, I specifically chose these examples. We have a ketone, we have a CO2. We would have a different molecule for an ester example, but I think these two examples are enough. So in the ketone example, we have some carbons, and well, look at this. We can use ATP to actually create carbanion. It's going to attack this carbon right here, attach itself, and we're going to be having this molecule. So this is citral coenzyme A. Okay, so for some reason this picture didn't show the usage of ATP. But that's okay. So to recap, we have the bond formation. So this is actually bond formation using the energy of ATP. Okay, so typically you're going to be working with the carbanion. You can find the carbanion from ketones, esters, or carbon dioxide. You have to combine that with ATP, and you're going to be creating a bond between the carbanion and the molecule that is going to be interacting. And typically you're going to have ADP and phosphate ion as a product. Okay, so that concludes the cellular metabolism lecture. Okay, so these actually tie in directly to glycolysis and carbohydrates. So hopefully this allowed you to understand what the concepts behind the material. I hope you do well on your exam, and thank you so much for watching the video and spending some time with me. Hope you have a great day and that you continue to do well in your classes. Thank you.